I'm recording. My job is okay. Everybody's ready. So I think I know everybody here. I mean, we haven't necessarily interacted directly much, but for those online who don't know me, I'm Leslie Roll-Buckland. I'm a research assistant professor in the department. I'm also the other person who teaches physiology, and usually I teach the online version. So hopefully that's a nice change of pace, alternating between Troy and I. And so today we're going to talk about locomotion and muscle function. And I'm really insisting on muscle function because that's the physiological aspect. Let's see. Oh, and that was supposed to be a little video of a bee flying, but it's not working for some reason. It's okay, you've seen bees before. So first we're going to define what is locomotion. It's just the movement from point A to point B. And so by that, I mean either small trips or long migrations. It does not matter. It's just that what will involve muscles and movement from point A to point B. So there are many reasons, many important reasons why insects need to move, and for that matter, other animals as well, to find food and water, obviously, to find shelter and protection from predation, which this fruit fly failed to do, obviously, and also finding a mate. And although I'm not going to talk about behavior, and I don't know if Troy's covering behavior in other lectures, but it is linked to it. And it's also a point I would like to make. A lot of the systems that you've already covered, that you're going to cover in the next couple of weeks, they're all interconnected. So what you've learned 3 weeks ago is going to apply here too. So behavior involves decision making, so nervous system, it involves locomotion, movement, muscle function. It involves communication, so especially for finding a mate, communicating pheromones with the opposite sex, and then the male going to find the female. These are all interconnected. So that's just a point I wanted to make here. Okay, so just a little structure of what we're going to talk about today. So first is more anatomical, an anatomical point about muscles and their structure, but also the structures that are surrounding them. And then the second point would be the connection with the nervous system. So it's more how muscles work, how contraction occurs, and how it's regulated. And then we're going to make a point on flight muscles because they're so unique to insects, because even other animals that evolved flight, like bats or birds, they have very different structures for flight, especially. So it's going to be really unique to insects, what we're going to talk about at the end of this lecture. And so here are some of the structures. So just to put in context, we're going to break down the muscle to its functioning unit, and even the proteins that are involved in muscle function. So the first thing is there's a certain vocabulary that's involved with muscles and muscle function. So the first one is from the Greek, it's sarco, and the other one is from the Latin, is myo. And you may not even realize that you already know some of these words, like sarcoma, which is a cancer of muscles and bones that are associated with muscles. But there's also structures of the muscle cell itself. The sarcolemma is actually the membrane of the muscle cell. The sarcoplasm is the cytoplasm of the muscle cell. And this one, very transparently, sarcoplasmic reticulum is the endoplasmic reticulum of the muscle cell. But if you say the endoplasmic reticulum of the muscle cell, it's not wrong. It's just that there is a special name for it. And that comes from the fact that cells were studied mostly in vertebrates, actually, to begin with. and they were studied separately as a cell structure from other types of cells. So they have that vernacular associated with it. And trying to find examples of myo, I guess it's not as much used. Well, there's myosin, that's one of the proteins that's involved in muscle function, and myopathy, that's diseases of the muscle, to give you a couple examples. Let me know if I'm going too fast. If you're taking notes or something. But yeah, so also something that's important to realize is that we're talking about muscle structure and function in locomotion, but muscles don't just control locomotion. Obviously, there are other events that are more involuntary that are also controlled by muscles. It's just, in some cases, it's different types of muscles. So like, for example, oviposition will involve some muscles, and moving antennae as well, digestion. And so that's going to be just the same as for us. Digestion is fully involuntary, and there are muscles along the digestive tract that are involved and that will contract and help the food move through the digestive tract. And in insects, it's the same thing. So there are a lot of things that are conserved across all animal groups. So that's kind of nice, because if you understand some of those basic functions like muscle contraction, the nervous system functioning too, it's pretty well conserved across animals. So there's also a couple differences, obviously. I'm guessing that's because of their smaller size, but insects have just a smaller number of muscle cells than vertebrates. And yeah, to go back to that idea that they are voluntary and involuntary movements, for these two types of functions, there are two types of muscles: the visceral muscles that control peristalsis, so like digestion, involuntary movement, and skeletal muscles that are involved in locomotion. So the ones that we're really going to focus on today. And so here on the right, it's an example of the thorax of a strepsipteran, but it's probably very similar to other insects. And I highlighted two, with two arrows, a structure that's called a tonofibrillae. That's what allows the muscle, the skeletal muscle, to be attached to the skeleton. So here, it's the exoskeleton, obviously, in insects. But in vertebrates, it's going to be attached to, the skeletal muscles will be attached to the endoskeleton. So like I said, that idea is conserved, it's just a different type of skeleton. So the tonofibrillae, they are also impervious to molting fluids. So during molting, their integrity is conserved. And so that allows the muscles to stay attached. So even when they're molting, when they are shedding their old cuticle, insects can still move. They're very vulnerable. So if they need to escape, they still can do that. So that's pretty important for their survival. Whoops, well, that moved. That's okay. Just following along to make sure that I didn't forget anything. So yeah, we've seen that. Where am I? The structure, that drawing in the slide a couple slides ago. And so that's just to put the muscle back in context and break down the different parts of the muscle. Here on the right, from the organ to its really smallest contractile unit. And the first, I guess the most important part that you need to understand is that the muscle fiber, which is the cell, the muscle fiber is it has an excitable membrane, an electrically excitable membrane, just like neurons. So you should already have an understanding of how that works. You've seen the nervous system, right? Already. To me, that's the most important system. So I I knew you already had seen it, but it's going to work exactly the same way. There's depolarization, hyperpolarization. It works exactly the same way. And we're going to talk a little bit more about that in a couple slides, but it's also because there is a communication between the nervous system and the muscular cells and the muscles in general. I put this here. It's kind of out of place, but I'm not sure if it's going to come back at all during the rest of the presentation. So a muscle can only contract. It can't lengthen on its own. It can relax, but it can't extend on its own. It's just through the work of an antagonist muscle, so a muscle that does the opposite function. So I thought of an example that's pretty straightforward. When you extend and bend your arm, your biceps and your triceps are working oppositely. So I think that's what it means. I mean, I know that's what it means, but it's just to say that a muscle can only contract. It can't extend on its own. Just in case we can't go back to that, I thought it needed to be said. There's also a difference between visceral and skeletal muscles that we might not go back to either. It's just that typically skeletal muscles tend to have multiple nuclei, where visceral muscles tend to have only one nucleus. That could be a good question on the exam, true, false, an easy one. And yeah, so if we look at the structure, it's very, I mean, you probably have already seen that, but muscles are very striated. And that's because under the microscope, the light is reflected a certain way by the different structures. And so that gives that idea of striation. And so if we break it down from the organ, then we have the muscle fibers that constitute the muscle. And each of these fibers is actually one cell. So they are pretty long cells. And that's probably why they have, some of them have multiple nuclei. Then if we look at those muscle fiber or muscle cells individually, you can see that there is a succession of smaller units, smaller structures, and this succession put together is called the myofibril. And if we look at this smaller unit here on the bottom right, it's called the sarcomere. Sarco, again, that means muscle, and that's delimited between those two light gray structures here. But that is really the unit of contraction. That's the movement of those proteins relative to each other that will allow the contraction. And so I mentioned proteins. There are two major proteins here, actin and myosin. So these are the most important, but then there are other proteins that are involved in the contraction, and we'll get back to that in the next slides. Yeah, so that's just another view of the sarcomere. Two other views actually. So this one is kind of similar to what we saw in the previous slide here. So on the left, it's very simplified. This one is meant to be a little more realistic on the right. And so actin is that protein that is found in lighter gray here. Those indentations here, if you want. And that corresponds to that red double helix protein on the right-hand side. And so these are the two states of the sarcomere, either at rest, relaxed, or contracted at the bottom. And so these are two different views because I thought maybe one would be more helpful for you to understand how it works. But basically, you can see that the myosin has heads that connect to actin, and that's really where the contraction will be initiated and will work. And so there are different delimitations here. And so some of them, they got their name because of the way they reflect the light. So the I band, for example, is from isotropic, and A band is from anisotropic. It doesn't matter, but just to tell you that it doesn't come from nowhere. There's a reason why. And then the M line is probably because it's median. Honestly, I didn't double check, but if there's a way to remember it, maybe that's what it is, even if it's not what it stands for. But so within the sarcomere, there are different regions. And so each of them will be, some of them will change size during the contraction. Some of them will stay constant. But so here are those different structures. So first, a sarcomere is delimited between two Z lines. But you need to think that it's not a line, it's actually a disk because it's a 3D structure. I don't know if that's going to be visible in the recording, but so what you see is this, whatever. But really, it's more like a cylinder. Right, and that would be Z-disk. And everything is in 3D, so that would be actin, myosin with all its heads. Whatever, you get the idea. It's a 3D structure. So Z line is also Z-disk because of that 3D structure as a cylinder. So that's the easy one because it delimits the sarcomere. Then another easy one, the M line in the middle, M for the middle, that's easy. And across from each, basically the M line splits the H zone in half. And the H zone is where there's only myosin. So there's no overlap between myosin and actin. So there's really this middle part. So you can imagine during contraction, I mean, you don't have to imagine, it's right here at the bottom. This H zone is very small, right? The H zone is comprised within the A band, and this A band is where myosin and actin overlap from each side, on each side of the M line and on each side of the H zone. And so this one doesn't, since it goes the length of myosin, it doesn't really change its size during the contraction, if that makes sense. And then the last structure is the I band. So this one is only where actin is alone. There's no overlap with myosin, but it's going from one sarcomere to the neighboring one. So like similarly, it only shows one side. So that's the Z disk from one sarcomere and the I band. And that would be like myosin comes here. Yeah. So that's the I band. So then myosin will come here. So that's where it stops. Does that make sense? So then when it's contracted, it's really small. As you can see here, the I band is going to change size as well. It's going to be very small. So which ones are going to change in size during contraction and relaxation? H and I. So I, yeah, I just said it. H, yeah, we did say that it's where myosin is present alone, and since actin will be brought closer on each side, then H is going to be smaller. It's A that doesn't change. Okay. And that allows me to make another remark about the difference between skeletal and visceral muscles. The sarcomeres of the skeletal muscles is smaller, about 2 microns in length. And in visceral muscles, it can go up to 10 microns. So typically, visceral muscles tend to be longer. So let's go back to the protein, since I already brought them up with this horrible drawing here. But we have the 2 main ones, actin, myosin, and myosin has heads. What's important about those heads is that they all have an ATPase activity. And some of you might be like, oh no, ATP, I know what that means. Yeah, there's a little bit of electron transport chain, mitochondria, all that stuff that everybody likes. So yeah, muscle function requires energy. There's ATP consumed, so that means there's going to be needing, it's going to be needing ATP to be brought into the cell. So that's going to be important in a couple slides that you remember that. But so yeah, they all have an ATPase activity, and so they will be able to hydrolyze ATP to use the energy of the hydrolysis to move. So actin, myosin, there's also two other proteins that are associated with actin that are called tropomyosin and troponin. So troponin, you'll see, is made of 3 subunits, and that's going to be important in contraction. And yeah, nothing that you need to remember. And then I didn't say it, but there are also other proteins that are involved, and it depends on the muscle. It depends on the location in the body. And so they are called accessory proteins. Some of them are structural, like paramyosin. It's part of the thick fibers. Then titin is a more filamentous protein. And this one is associated with kinase. So I don't know if every time you hear kinase, you think phosphorylation. That's what it does. And phosphorylation is used a lot in cascade and signaling within the cell. So that's what it's associated with. But it's also a protein that provides resistance to stretching in the sarcomere, so it doesn't lose its structure. Projectins are other of these proteins are present in A bands of synchronous muscles and I bands of asynchronous muscles. So it must sound a little nebulous right now, but it's going to be clear to you what are synchronous and asynchronous muscles when we talk about flight. Then flightin is another protein. Pretty transparently, it's involved in flight muscles. It's also present in thick filaments. And then I should have put these ones first because that's going to be very important in muscles. There are calcium binding proteins. So troponin C is one of the subunits of troponin. That's one of them. It will interact with calcium ions. Calmodulin is also another one. And there are also other calcium binding proteins in the cell, in the muscle cell. And calcium is really key in contraction. Then arthrin, it's also present in flight muscles. It has a role in stretch activation. And chitin, it's just another of those proteins that provide stiffness for stretch activation as well. And same thing, it's going to be a little bit more clear towards the end of the lecture because they're involved in flight muscles, so we're going to talk about that a little more. Try not to go too fast. These slides are a little heavy. You'll have them, so don't worry about taking too many notes. So here is more what I'm excited about. What I really want to talk about is more the functioning, how things get in motion, how locomotion works, and basically how the muscle contracts. And so here is a very simplified version, a simplified vision of how the muscle contraction is initiated. It is the neuromuscular junction. So here you have the end of the axon from the neuron. So it's sending the nerve impulse to the muscle cell that's connected to it through chemical communication. So in a synapse-like connection. Actually, I don't know if it's called a synapse or if it has a specific name, but it's basically what it is. And so there's going to be movements of ions, of neurotransmitters that will bind to the muscle cell to send the message chemically that it needs to reinitiate an electrical message to go through the muscle cell. And so just like in the nervous system, there are going to be movements of ions through ion channels, ion pumps, that will allow the depolarization of the membrane. So there are going to be two main events that will trigger the muscle contraction. First one, as what I just said, the muscle depolarization or the membrane depolarization via the reception of the nerve impulse. So that triggers the contraction. And then it implies the release of calcium within the sarcoplasm. And the calcium concentration, the calcium release will tell the muscle when to contract, but also as long as the calcium is out in the sarcoplasm, it will tell the muscle how long to contract for. So that's going to be important. So first message here, the muscle needs to stop contracting at some point. So there's going to be a release of calcium ions, but they are going to be, it's going to be necessary to put them back where they came from so the contraction can stop. Because you can imagine continuous contraction, it's not a good thing. And yeah, so this is on the left-hand side. Do you remember the muscle structure? So like something like this, the striation, whatever. So that's a muscle. This is a cross section, so that means we cut it that way. So what we're seeing is that slice here facing us. So there are structures here that are going to be important for the contraction. So the myofibrils, we talked about that already. The tracheae, there's a nucleus here, mitochondria. It's not super clear here, but they are there. And T-tubules and vesicles of the sarcoplasmic reticulum are the two that I highlighted here because they are the two that will start the process. So T-tubules are actually invaginations of the membrane within the muscle structure, how can I say that? Yeah, within the muscle. And so that will really bring the electrical message within the muscle to really communicate its depolarization time, its contraction time. And the membrane has contact with the vesicles of the sarcoplasmic reticulum, or reticulum, endoplasmic reticulum of the muscle cell. I remind you, it's exactly the same thing. It's just the name that's different. And so what happens is that they will sense, and I use very clear quotation marks because they don't really sense, but they are sensitive to change of potential, I guess. So yeah, they sense it. Let's keep it that way. So the electrical potential will propagate through the membrane that was previously more positive on the outside, more negative on the inside, at resting state, it will become less negative on the inside, and calcium will be released from the sarcoplasmic reticulum. So that's what starts the process. There's depolarization of the membrane and release of the calcium ions from their storage. And then the relaxation, the return to resting state is just calcium going back into the sarcoplasmic reticulum through a calcium pump, because just like everything, ions, or like systems that you've seen before, ions passively migrate through channels, through pores, along their gradient. So this will be against this gradient. So energy needs to be used to bring it back into storage. And so that's why a calcium pump is used. It's just an ion channel with an ATPase activity that is able to use the energy of ATPase to bring back in an ion against its concentration gradient. And then, the membrane is repolarized just the way it was before it all started. Yes? As long as calcium is out? Yeah. Inside the cytoplasm. Yep, yeah. So that was the structures. Now let's look at the proteins and how they behave during the contraction. So remember, we were at the cell level. Now we're at the sarcomere level. So it's a unit within the cell. So I actually inverted the colors without paying attention. So my actin is yellow here, and myosin is kind of reddish purple instead of being yellow and the other one was red. But so, I mean, you recognize the double helix of the actin protein and the myosin head here. I only represented one just for simplicity. But imagine there are a lot going on, like here, here, here. And then that's just one pair, but then you have a couple more within the sarcomere on both sides. And then there are other sarcomeres next to that. And so basically, so we have our other proteins, tropomyosin in green here, that goes around the actin, and troponin with the 3 subunits. So the 3 subunits, I don't think I mentioned it. So we talked about one that's associated with actin. And so this one is called the subunit I. Doesn't quite make sense. But the other two will make more sense. So I might add it to the side, but I'll also give you notes. So for each slide, I'm going to have some sort of, this is what I said in the slide. So I know I have an accent, I can't get rid of it. And sometimes it's not easy to understand what I'm saying. So I just want to make sure that you have the words written down in front of you so you know what I said. Oh, that's how it's spelled. So this one binds to actin. Then you have a troponin T that binds to tropomyosin. So that green myosin, the green protein. And then you have C. This one binds to calcium ions. So these two make more sense. I don't know why they didn't call it troponin A, honestly. So I cannot give you an explanation. So that's structure. Now let's go back to what happened before that. Calcium ions were released in the cell. So we're in the sarcoplasm here, the cytoplasm of the muscle cell. Calcium is in abundance in the cell, and so troponin C is going to bind to calcium. And that will trigger a change in conformation of troponin C, troponin T, that will rotate along with tropomyosin that will rotate. And doing that, it releases or it unveils binding sites on actin for the myosin heads. So then the myosin heads will bind to those binding sites on the actin. And you'll also, maybe that was a little premature, the myosin head was bound to an ADP, but it will release its ADP and then bind an ATP. And that's where it will be able to hydrolyze ATP into ADP and phosphate. And the energy of the hydrolysis is what will make the head move. And so basically how actin and myosin move is relative to each other. And it's kind of like pulling a rope, you know, if that makes sense. And actin would be the rope and the myosin head would be the hands. So as they release the energy of the hydrolysis, they can move and move actin along. Does that explain it? So again, it's just one head here, but there's quite a few working on at the same time in the same movement. Yeah, and that's a cycle too. It's not just one time. It depends on the muscle. It depends on the innervation of the fibers. It depends on, we'll talk about the type of innervation too, but it's not just going on one time. There could be multiple contractions, or like one contraction and release, and another contraction. But yeah, so just to summarize again, to make sure that it's clear. So myosin head is bound to ADP. So adenosine diphosphate. That means it has 2 phosphates in its structure. And then troponin C binds to calcium ions. And this will trigger a change of conformation of the proteins that are wrapped around actin, which make it possible for myosin heads to bind to actin. Then it can release its ADP and bind an ATP. So yeah, it's going counterclockwise. To think of mentioning, so it's going counterclockwise. And so the ADP is released. It binds an ATP. ATP is adenosine triphosphate because it has 3 phosphates. And it can hydrolyze this ATP into ADP and phosphate. And the energy that is released allows for the movement of the head along the actin. It detaches from the actin. Here. Here. So you can see when the binding sites are shown by the conformation change, it finds a way to bind to it. Like it recognizes there's some sort of affinity, and so it can bind to actin. It's attracted to it. And then once it hydrolyzes its ATP, it will release. And then it repeats that. So that's how the rope is pulled, if you want. I know it might not seem clear. There is a video where I got that image from. Where is it? I don't know if I have the link here. No, so it must be at the end, but that's from a video. This view on the right is from a video, and I have the link at the end. So if you want to check this out, maybe it's explained better than how I explain it. And you know, what works for some people doesn't always work for everybody. So I explain it a certain way, but you might need to find another source. Okay. So I mentioned innervation. So that's kind of implied that to be able to receive a nervous impulse, a nerve needs to make connection with the muscle and the muscle cells. So that's what innervation is. And Yeah, I'm trying to kind of get the main idea from this slide where there's a lot of text. There are different types of neurons that can connect with muscle cells. Some of them are called fast neurons, some of them are called slow neurons. And so depending on the muscle, depending on the fiber within the same muscle, they may be innervated by different types of neurons. And fast neurons cause a rapid contraction. Slow neurons is more incremental. So here on the bottom right, that's the difference on how fast and slow neurons work. And basically, that's the fast neuron is more like the all or nothing that we've seen, or that you've seen with the nervous system, how it works, the depolarization gets to a point and then it repolarizes, then going into hyperpolarization. But for slow neurons, it's more like a gradual potential. So there is some sort of addition up to a certain amplitude, and then it will depolarize. So it takes a little bit longer. So that's why it's slow. And there's also repeated firing here, so twitching. So for some muscles, it might be important to twitch instead of just being contracted all at once. So this one is, the fast neurons is more an all or nothing response, and this one is going to be multiple firing in gradation until it gets to a certain point where it will repolarize, sorry. Yes.
Can you remind me what you said innervation was at the beginning? You
Yeah.
sort of grasped it from the slide.
Yes, it's the connection between neurons and muscles. When we say something is innervated, is that there is I'm trying to find another way than saying there's a connection, because it's very vague. Like they get messages from neurons.
Okay.
There are tissues that don't have neurons, but muscles are not one of them. Yeah, I don't know how to explain that any other way. Oh yeah, so that has nothing to do with the speed of how the message travels through the neuron. Is just the contraction itself that gives it a name, fast or slow, the type of contraction.
So it's not either or. Like there's not one insect that only has fast or slow. It's just
No, yeah.
It's a twitch or a.
Yeah, I think it depends more on the muscle. So insects can have the two different types. And yeah, we're talking about skeletal muscles here, just so we're clear, not visceral muscles. And I think it's well explained in the book. You know, when you summarize information, sometimes it doesn't translate very well. So if you're still not sure, if you have questions on Wednesday and I can't answer, make it clear. If you go back to the book, maybe that will help. Not that I'm releasing responsibility. No, I want to be able to answer your questions, but if my answer doesn't help, there's always another resource. Okay, so we talked about that junction between neuron and muscle. I mentioned neurotransmitters. So that's what we're going to talk about here in terms of regulation, nervous regulation of the muscle contraction. So in nature, the nerve impulse is excitatory, right? And so there's going to be inhibitory neurotransmitters that are released to give the message to the cell, basically, okay, this is not an excitation. This is not going to be a contraction. That's going to be relaxation, or just don't do anything. So the main inhibitory neurotransmitter is gamma aminobutyric acid, also called GABA, that acts postsynaptically to affect muscle response to excitatory neurons. So here is the structure of GABA on the right. So opposite to that, you talked about acetylcholine in the nervous system as the main excitatory neurotransmitter, but there's another one that's called L-glutamate. So until recently, we thought that it was only present in insects, or like in invertebrates at least, that vertebrates did not use the same excitatory neurotransmitter at the neuromuscular junction, but actually it's been found recently that they do, or that we do too. There's been evidence in a lot of model organisms like mice, zebrafish, frogs. Yeah, different developmental stages. So it's not just in adults, it's not just in young, it's across their life stages. So it's kind of new. We thought there was a difference between how neuromuscular junctions worked in vertebrates and invertebrates. It might not be as different as we thought. But so this one is it's excitatory. So it will give the information to the muscle cell, to the muscle membrane to depolarize, to continue sending an excitatory message on the muscle membrane and to make it contract, basically. That's the idea. And so it uses the T-tubules that are those invagination to send really the message deep into the muscle. So then there are other molecules that affect neurotransmitters. So they're called neuromodulators. So they can be either excitatory or inhibitory, depending on the conditions. But so for example, octopamine is one of them. So I'm not sure if you have already talked about it, but I'm pretty sure you're going to talk about it next week in communication because it's involved in how fireflies can produce light. So that's kind of cool. It does a lot of different things. And so it is involved in muscle function. It is involved in light production, so in communication. So that's pretty interesting. But so what it does is that it can synergize L-glutamate. So it's going to reinforce its excitatory abilities by releasing cyclic AMP in the cell. And cyclic AMP, I'm not sure if you have seen that, but I think you have probably in the endocrine system and potentially in the immune system, because that's a pretty common signaling molecule. So it's just used to create communication within the cell, between the different parts of the cell, and it can even increase or decrease the transcription of some genes, depending on when needs to be activated or inhibited. So that's pretty variable depending on the function. It's very vague too, I'm sorry. But yeah, maybe by next week or next session, I can have some examples. Well, I guess that's one of them in flight muscles. So like I mentioned, muscles need a lot of energy and flight muscles even more than other muscles. So there is a connection between metabolism and muscle function. And one of these connections is through lipid metabolism. Lipids are a source of energy for muscles, for flight muscles particularly, as opposed to maybe more glycolysis in other tissues. But so it helps. Octopamine releases CAMP levels or increases CAMP levels that helps produce adipokinetic hormone that will be involved in lipid metabolism to release energy for flight. So there are multiple steps that can be activated by the release of octopamine. Oh, I don't like this slide. Yeah, they're just other molecules that are called myotropic peptides. So peptides, that just means proteins that have less than 50 amino acids. So they're very small. And they are just other modulators, just like octopamine can modulate the action of neurotransmitters. Those myotropic peptides can also modulate, they can modulate stimulation or inhibition. So for example, proctolin is one of them. So this one has been described in the excretory, the digestive tract, the never gonna make it, excretory organs of insects and visceral and skeletal muscles. And it's only a pentapeptide, so that means it has 5 amino acids: arginine, tyrosine, leucine, proline, and threonine. There are other examples, the cardioacceleratory peptides, or CAP. They control muscles that produce heartbeat. So again, that's another type of muscle function. It's not locomotion, but it's another muscle function. And it's indirectly connected with wing beat or with locomotion because it's connected to wing beat, because heartbeat will influence wing beat. Yeah. And the other two, it doesn't matter. It's not locomotion, so I don't really know why I put this here. It doesn't matter. Yeah. So yeah, we can finish next week. If you're really, I can't do this anymore, I need to get out, just let me know. But otherwise, I have a couple more slides and a couple more things to tell you. So for example, where the energy for contraction comes from. So I know a lot of people see that they have a very strong reaction, almost allergy to the Krebs cycle and electron transport chain, and just hearing the name mitochondria, they're like, ah. But I don't know. I think once you kind of get the main steps, you don't need to know step by step what's happening, at least not for this class. Maybe more for like digestion, metabolism. Have you seen that already in previous lectures? Do you do digestion? Did that? No? No? Come on. Yeah, I found a video that's kind of fun. It's a little dumb. Like some students created a song about how this works so they can remember it easily, the main steps. I mean, if you want to watch it just for your general information, that could give you some keys to even for other classes. But I thought that was pretty fun. I can send you the link. So where am I? 24. Just want to make sure I'm not forgetting. So yeah, I said we need a lot of energy to make muscles work. And when you work out, you feel tired, you need sugar, you need to replenish your energy. Well, think flight, running, walking in insects is the same thing. So they need to find energy somewhere. And one of the energy sources that we've already talked about is ATP. That's produced in the innermost membrane of the mitochondrion. You know, there are 2 membranes, kind of a weird organ or organelle. So there are 2 membranes. The inner membrane is where the electron transport chain occurs. You see that it needs oxygen to work. And the endpoint is that it produces ATP by transporting protons across the membrane. And then this ATP is used as energy currency of the cell. So 2 things to notice. ATP is produced, but it needs oxygen. Okay, I'm saying that because, so ATP is produced in the mitochondrion or mitochondria for plural. And oxygen is provided by tracheae. So that's the respiratory system of insects, is different from our respiratory system. It's, the tracheae are connected to spiracles, so they're connected to the cuticle of insects. And they have projections within muscles. So these can even project into tracheals, like you can see on the right side. Tracheals are just projections of tracheae, and they will make direct contact by invaginations into the mitochondrion or into the muscle fiber, in contact with the mitochondrion to really be able to provide this oxygen that comes from the air to the mitochondrion so it can produce ATP. So there is a link between oxygen supplies, metabolic activity, energy production, in other words, and like everything muscle related, flight is going to be very energy demanding. So there are also other structures that are involved here to provide even more energy during flight. Insects have, what's it called? I'm trying to find it. Air sacs. Yeah. So yeah, the air sacs can change in volume to accommodate the oxygen needs. So during flight, they can inflate to provide more oxygen. You did? Okay, that's good. So there's a contraction. I don't know how I wrote it. It doesn't quite make sense, but there's an abdominal contraction that helps move the air. That's kind of like a pump. It moves the air through the system so it can provide more oxygen to the muscles. Okay. Yeah, I said that already. Yeah, it's a pumping action through the tracheal system for the air to circulate more, faster. And mitochondria use oxygen to produce the energy required for movement. Okay. So now that's the end of the lecture soon, I promise. We get to the point where we're going to talk about flight, which is like nothing else that exists in other animals. I mean, flight exists in other animals, but this type of flight exists only in insects. So there are two types of skeletal muscles that are involved in flight and also other functions. We have synchronous muscles that are the predominant skeletal muscles and asynchronous flight muscles. So these are just the two types, synchronous on the left, asynchronous on the right. Okay. So they are named this way. There are two different categories because it's based on their mechanisms of regulation by the nervous system. So synchronous muscles are most of the skeletal muscles in insects. There are the muscles in the leg, like you can see on the right here, in the limbs at least. And so they are very important in locomotion, but they're also present in some flight muscle, or they are representing some flight muscles in some insects. Basically, they are called synchronous because they contract in synchrony with the nervous signal from a motor neuron. So they get one signal, it will lead to one contraction. That's it. There's one electrical signal, one contraction, and it's going to be limited in frequency because of the refractory period. Remember that hyperpolarization before going back to the resting state? That's going to limit the contraction. So it's also going to limit the frequency of wing beats in flight muscles, in synchronous flight muscles. And they have a lot of sarcoplasmic, they have a big sarcoplasmic reticulum. It can take up to 1/3 of the cell. So that means there are a lot of calcium ions are released and also recaptured. So it's pretty fast. But these muscles contract only, or at least most of them only contract to 50% of their capability. And so they are considered primitive flight muscles, not because they are present in primitive organisms, but because they are the first to have appeared in evolution. So it's not a negative term. It's just that was the first version of the same structure. And the paper that I assigned for you to read before Wednesday for our discussion is about particularly what happened in Lepidopterans. I'm not going to say more because I want you to read it. So that's a simple explanation. It appeared first. It's present in some of the older groups of insects like orthopterans and odonates, but also in Lepidopterans that have appeared more recently. So what happened there? And that's kind of outside the locomotion theme. But synchronous muscles are also found in other structures like the muscle that allows sound production in cicadas. So that has more to do with communication than locomotion. And that's the highest known frequency of contraction in a synchronous muscle. And that's in this muscle that the sarcoplasmic reticulum occupies a third of the space. So just an example that's a little bit outside of the lecture, but that could be interesting. So the other type of muscle is the asynchronous muscle, or fibrillar. It's also called fibrillar. And so in this case, it is found in wings, or to make the wings function in dipterans, coleopterans, hymenopterans, and some hemiptera. So they are, some of them are pretty old groups, but some of them are the most recently evolved ones. And it corresponds to the evolution of a smaller wing, and that allowed those insects with smaller wings to occupy niches that were not occupied by larger insects or insects with larger wings. Like imagine odonates have really big wings, lepidopterans have very big wings. And so like fruit flies, very small, compact organisms. And so they have been able to occupy those areas that were not available to other insects with bigger wings. These muscles allow a high frequency, and they can actually receive 5 to 25, they can produce 5 to 25 contractions per nerve impulse. So we're not one-to-one like with the synchronous muscle. One nerve impulse can allow 5 to 25 contractions. So you can imagine that the wing beat is going to be a lot more accelerated. They're going to be able to bat their wings a lot faster than those bigger insects with synchronous flight muscles. What's kind of interesting is that this evolved independently multiple times. It's not from a common ancestor. It's an adaptation that evolved in different groups in different ways, but the result is the same, and we call that a convergence. And there are a lot of mitochondria, a lot of myofibrils, short sarcomeres, even shorter than in other skeletal muscles. So just more energy for the increased wing beat. They also have a reduced sarcoplasmic reticulum. So the calcium concentration is not going to change as much. And somehow it saves energy and oxygen. I'm not exactly sure how that works, but that's what I found. So, and so there's a link here to go to the article I talked about that talks about the evolution of asynchronous muscles and synchronous muscles in some insects for flight. And yeah, I don't know, it's going to be, I don't know, maybe I should keep that for Wednesday because I don't want to make you go crazy. That was too much. I don't understand anything anymore. I'm too hungry. So yeah, we could keep that real quick, 5 minutes at the beginning of the next session on Wednesday. But so what I wanted to say is that there are a couple videos on the contraction that I thought would help. If you want to look at the click mechanism, what I'm going to talk about next on Wednesday, it's going to be a little easier for you to understand. And yeah, that's the chapter in the book if you have it. And that's the article that I would like to discuss on Wednesday. So that's a simplified version, but that's the full article that was published in Nature. It's about when asynchronous muscles evolved. And so I just want to have a discussion about it, what you learned from it, how they found out. And there are robots in this, so that might be interesting for some of you. So yeah, let's do that. So about, okay, 10, 15 minutes, we can talk about what's left, the click mechanism in flight. We can talk about the lab for the graduate students, because that's important to kind of share with everybody applications of why I'm talking about this, because yeah, why? What are we gonna do with that? And so the undergrads can understand, and people online too, and some other techniques. And then we're going to talk about that. Sounds good? Yeah, do you have questions? Yeah, prepare questions for Wednesday if you don't have any today. I know that's a lot. Kind of tend to dump a lot of information, and then you kind of have to regroup and what's the main information here? Mila?
He canceled last lesson.
Oh, really? That's okay. We can still talk about that. And good reason to talk about that. Okay, so if I say yes, is it going to send it to the cloud? I'm scared now. Yeah.
I never watched the part.